Good afternoon, everybody, and welcome to this session on nitrosamine mitigation. So in the next 15 minutes, I will quickly go over the nitrosamine issue history. I will then share some information about nitrite scavengers as nitrosamine mitigation agent, and specifically our learning on ascorbic acid as a mitigation tool for nitrosamine formation, and then we'll conclude. So nitrosamine are everywhere. They've been known and studied for decades. And the first link to cancer was actually described back in the 50s. So the FDA has started really scrutinizing the topic much more recently with the discovery of nitrosamines in sartans back in 2018. And since then, they've been evaluating and regulating it. So today, the need to evaluate, measure, and mitigate is there to stay. And so all new product being developed will have to address this early on in the process. And while the limits of nitrosamines in different product will evolve as the evidence for safety and carcinogenicity of nitrosamines accumulate, the need for control and mitigation will stay. So and nitrosamines are formed by a chemical reaction between a nitrosating agent and a secondary or vulnerable secondary or tertiary amines. And a number of these compounds have carcinogenic potentials. So the conditions for nitrosamine formation can be met at many steps during the drug product process, from the drug product themselves, so the API, but it's also the excipient, potentially it's the solvent, the water, the manufacturing itself, the packaging, and the storage. So it can all create conditions for the generations of nitrosamines. Now, many strategies can be used to deal with the formation of nitrosamines. For example, strong qualification program for all compounds, using making sure to keep the level of nitrosating agents low. And today we're going to focus on one of those strategies for mitigating the issue, and it's using nitrite scavengers to reduce or stop the nitrosamines from being formed. So there are many nitrite scavengers able to do the job, and some of them are listed on this slide. But they're not all equal, however. So some of them are not on the inactive ingredient database, which of course make them not as attractive. And others, while they are on the inactive ingredient database, they don't work necessarily in solids. They might work in solution, or they might be sensitive to a variety of environmental compounds and not be appropriate. So, but two of them, in our experience, have shown consistently good results, and those are ascorbic acid and alpha tocopherol. So how do these two molecules block nitrosamine formation? Well, nitrates, which is often found as impurity in excipient, will form those nitrosating agents like nitrous acid, and they will further transform and react with the vulnerable amine being present. And both ascorbic acid and alpha tocopherol can interact rapidly with the nitrate and the nitrosating agent being around, and in this way they stop the nitrosamines from being formed. So this is really a redox process, and they reduce the nitrosating agent to non-nitrosating nitric oxide, and without nitrosating agents, no nitrosamines are formed. Now, ascorbic acid is best for moderate to strongly polar environment, while alpha tocopherol has more affinity for less polar or non-polar environment. So they can be used really in different conditions, with ascorbic acid more suitable for from solutions, syrups, injection tablets and capsules, and alpha tocopherol mostly used for creams, ointment, capsules as well, though, and tablets. So what you see on this slide is a table representing results that were obtained by Dr. Nanda back in 2021. And that was very significant data for us because they looked at the efficacy of ascorbic acid and sodium ascorbate, alpha-tocopherol, ferulic acid, and caffeic acid, different nitrite scavengers, on the inhibition of nitrosamine formation in a solid drug product. So this was for us the first time that we were seeing in solid drug product the efficacy of those molecules in being able to bring down the level of nitrosamines under stress at 50 degrees C and 75% relative humidity. So this was very encouraging. And since then, a number of publications and presentations at meetings have reinforced the ideas that these scavengers are safe and useful to keep nitrosamine formation in check. So on the efficacy side, so the work of Omshak and colleagues, as well as the work by FDA and GSK, has added some data showing that ascorbic acid in particular works well in tablet form. And on the potential impact on drug absorptions, we've seen Dr. Chris Bode from Pharma on Lab Services, which has also shown no impact on bioequivalence of class 3 model drugs. And on the toxicity and safety side, of course, there's a long history for both ascorbic acid and alpha-tocopherol as for safe use in many different products. So we've now, internally extended those experimentation to really understand how well it works. And so in the rest of the presentation, I'm going to share some of this information. So first, we kind of took a look at ascorbic acid itself, the product. And what you see on the left side is actually a graph of the various particle sizes that you can see in different vendors of ascorbic acid. So already just from the particle size, and when you think solid dosage form and direct compression, this can have an impact. So you just know your vendor, know the particle size and what you want to work with. And on the right side, we also looked at the impurity levels. So all those different vendors and those different sources of ascorbic acids, while they are very good, they do have a variability in impurity level. And so you see here one that was from dsm-firmenich and in comparison with another supplier. But what we see generally, depending on the vendor, again, you will have some differences there. And those are things that are important for you to know when you want to make sure you have a reliable and reproducible source of excipient. So, and this is a picture basically representing those different powders and how they look under the electron microscope. So, in the first step, we actually prepared some tablets. And we wanted to compare really the activity of ascorbic acid at different concentration, really understanding if it really worked and if you added more, you would see an impact. So we manufactured some tablets either via direct compression or wet granulations with increasing amount of ascorbic acid, the ultra fine powder, so the finest powder we could find, from 0.25% all the way to 1% under high stress condition, 40 degrees Celsius and 75% relative humidity. And what you can see, first of all, you can see that at T0, so just immediately we make the tablets, we measure. And what we were looking at here, those are tablets without an active pharmaceutical ingredient in it. We were looking at the ability of ascorbic acid to decrease the amount of nitrate in the tablet, meaning really scavenging, doing the job that it's supposed to do. And what we find is already at T0, you can see the nitrate has been quenched and is not available to react, form nitrosating agent, and further down the line, nitrosamines. And at 7 days, once it's compressed, you still have the ability, you still see an impact, or further impact with the reduction of nitrate. And of course, so the higher the concentration, the more of an impact. But of course, there would be limitation as you move along. We would not recommend going any higher than 1%. So in the next step, we decided, okay, well, let's look now. We know for sure in solid dosage form, there is this ability to really quench the nitrite in the process and in the tablet. What about the API-derived nitrosamines? So in this case, we took an API, formulated it as it's normally formulated, and added some ascorbic acid, 1% in this case, looking at it immediately at T0, and then after 2 weeks under stressful condition, 50 degrees Celsius and 75% relative humidity. And what you see here is that you can see the increase in nitrosamines, so the API-derived nitrosamines over time, over those 2 weeks in the control tablets, which have no ascorbic acid. But with the addition of ascorbic acid, this increase is dramatically reduced. So this was very good for us to see. So we decided to extend this experimentation and looked at 2 other drug models. So again, different drug models, different formulation, different characteristics to see whether we could see that as well in those different drugs. So in this case, we are looking at a fluid bed granulation process, but you could also do it, and we're doing it right now with also dry compression to see comparatively how it does. And what we saw is that when you add the ascorbic acid, and here it was 0.5% ascorbic acid, and we looked at it over a longer period of time. We went all the way to 90 days in this case. And what you can see is the control is the blue line. You can see the increase in the API-derived nitrosamines over time, going all the way to 90 days. And when you add the ascorbic acid, and from the beginning, even after 90 days, you can see that the nitrosamines are not being formed. They're just staying like at the beginning. So again, this was under stressful condition, 40 degrees, 75% relative humidity. So this data really told us that really ascorbic acid is a good molecule when trying to mitigate or bringing down the level of possible nitrosamines being formed. So in the final step, or just to add, for those of you who are not familiar working with nitrosamines, with ascorbic acid, you may find that occasionally, discoloration can occur when you work with ascorbic acid. This is a reducing agent, and if there are amino acids present, it can lead to discoloration. This has been studied and described for many decades as well. But what we found is always, it's sensitive to oxygen and temperature, humidity, which is not an issue necessarily if you're coating your tablet. But if you are not coating your tablet, what we found in our cases is that using the smallest particle size was most effective. First it goes all around in the tablet if it's well mixed, and so you have less of an issue of a little discoloration here and there. So in conclusion, really for us, ascorbic acid, and I didn't talk about it today, but we've found, we also have some data on alpha tocopherol, but really are proving to be useful tools when delineating a strategy to keep nitrosamines below the established limits by the regulatory body. And using low nitrite levels, API and excipients as much as possible, of course, is the first step. But considering ascorbic acid and also alpha tocopherol as a safety net when you need one. So in our experience, very low level of nitrate scavengers below 1% can have a very significant effect. So even 0.25% can really help you stay below the maximum limits. We also recommend to go with the lowest particle size of ascorbic acid if you go with dry compression in particular, to ensure close contact with the nitrate sources and nitrosating agent. And if you have the capacity, of course, you can try different particle sizes and see what works best for your particular situation. And with that, I will take questions. for ascorbic acid in particular, so oxalic acid is the only specific degradation product of ascorbic acid, which is limited. So that's something when you add ascorbic acid to your composition that you can have as a control. But when the ascorbic acid react, you have dihydroascorbic acid, which gets produced and can be degraded all the way to 2,3 decatogulinic acid afterwards. But so that's the process of degradation, which is also very characterized, and happy to share some information about this if you need. So with tocopherol, we have not studied it as much, but again, it's also quite well characterized in terms of what happened. And there's no discoloration like this with alpha tocopherol. At least we've not observed this. Or it can maybe, depending again on the condition, the situation, and the other ingredients, maybe get a little yellow and not pure white, for example. So there's slight shade, but that you would not have this issue. But again, with alpha tocopherol, this is well good and well in hydrophobic conditions. So in creams, ointment, it might not be the ingredients for you to use in a tablet in dry compression, because you have, again, you're dealing with an hydro, a compound that's not great for aqueous conditions. Yes, so I mean, when you had ascorbic acid, so yes, you will have potentially dihydroascorbic acid or further product down the line. I mean, again, ascorbic acid is on the inactive ingredient database. It's been it has decades of characterization behind. So I think it's also from all the other the products that are already approved on the market, and see the kind of degradation product that have been looked at and are okay with the FDA. So that's a conversation with the FDA to have if there's more than just oxalic acid or concerns about the degradation product. But for now, oxalic acid would be the product that you would add to your analytical methods to make sure it's controlled. We made the tablets from the beginning to the end, dry compression or wet granulation, and then kept the product, the finished tablets, under those stressful conditions for the amount of time that I mentioned during the talk. MCC was used, yes, in one of those tablets. We used the formulation that's established for the API we were looking at. So those are drugs that are on the market that have a formula that is available. MCC was used. And so in those cases, 0.5% was largely enough. And again, so depending, I mean, you can calculate what, given how it quench and the amount of nitrate you expect from your MCC or from excipients you have in your formula, how much ascorbic acid you might want to use. But again, even at very low level, 0.25, you have quite an impact. So it is, I mean, for you as a formulator, do you want to completely remove the ascorbic, the nitrosamine being formed, or do you need to bring them down to a certain level? So during the lifespan of the shelf life of your drug, it doesn't go over the limits which have been set by the FDA. Last year we were trying to set that up with a variety of other scavengers to have really the comparison with the ascorbic acid we have actually in-house. And so we've not done that experiment yet, but what I can tell you, and I can share more information if you come to the booth as well, the work that Omshak did in 2022, and I recommend you check his paper. So he looked at 19 different nitrate scavengers in solution, and for some of them then in tablets after. And what he found was that, so cysteine is, but it's an amino acid. Again, it's not applicable to everything. Cysteine works really well. And you might not have necessarily the discoloration impact. You'll have other drawbacks. Propyl gallate is another one that can also work well, but again, it comes with even the source. Is it excipient grade? So you can have other issues. So you need to take a look at your formula, your particular drug, the potential issue with incompatibility between the ingredients, and choose the best. But the comparison of OPSHAC is really useful, at least for the aqueous kind of solution, because the format of the experiment didn't really was not really for me appropriate for testing like alpha tocopherol, for example, because it's hydrophobic. But yes, information there, but so cysteine, propyl gallate, I would say, would be other alternative. But again, it depends on the condition. Propyl gallate in the experiments of OMCHAC in 2022 was not working in tablets. So it worked well in his in vitro study in solution, but not so much in tablets. So